So here's our neuron. Here we have the soma, which is the cell body. These are our dendrites, and at the end are the receptors for neurotransmitters. Here we have our axon with our myelin sheaths and our nodes of Ranvier. Should I go? Here is our axon terminal. These are our terminal buttons. This is our synaptic gap. And these are our dendrites of our postsynaptic neuron.
Neurons are not always transmitting signals. The transmission of an impulse depends on the ionic gradient that exists across the axonal membrane. In the axonal membrane, the concentration of potassium ions is greater than the concentration of sodium ions. In resting potential, a pump pushes 2 potassium ions into the cell for every sodium ion it pushes out. This results in a net loss of positive charges within the cell. Therefore, the resting potential of a neuron is polarized because there is more negative ions on the inside than on the outside. In a human neuron, the negative charge at resting state is negative 70 millivolts. In response to a stimulus, and if the stimulus has enough intensity to excite a neuron, the cell reaches its threshold or above. The threshold is the minimum amount of stimulus a neuron needs to respond. It is at negative 55 millivolts. Action potential is an electrical charge that travels down the neuron. It is a change in the membrane potential that produces a nerve impulse. It is an all or none response. It does not fire partway. Tiny gated sodium ion channels open up and allow sodium ions to rush into the cell so that the cell is now more positive on the inside than outside. This is known as depolarization. Then comes repolarization. After sodium ions flooded the neuron, the channels close and potassium channels open. The potassium ion runs out. Now again, the inside is more negative as the original polarization. During the refractory period, sodium-potassium pumps are used to re-establish the distribution of ions. It pushes out 3 sodium ions for every 2 potassiums it brings into the cell. The charge actually goes below negative 70, and then it slowly lets more sodium ions back inside to reach negative 70 again. This allows it to fire again. When an impulse reaches the terminal buttons at the end of an axon, the axon releases a neurotransmitter into the space between the 2 neurons known as a synapsis. The neurotransmitters diffuse across the synaptic cleft and bind to the receptors on the dendrites of the next neuron, called the postsynaptic neuron. The neurotransmitters are released from the terminal buttons of the presynaptic neuron from vesicle sacs. In reuptake, excess neurotransmitters are reabsorbed by the sending neuron. Some signals are excitatory, which is like pushing the neuron's accelerator, while others are inhibitory, which push on the neuron's brakes. When the excitatory signals minus inhibitory signals exceed a threshold, action potential is triggered. Sometimes a neuron has supporting cells wrapped around its axon known as Schwann cells. These cells produce the myelin sheath, which insulates the axon. The whole axon isn't covered. The spaces between the myelin sheaths are called the nodes of Ranvier. Myelin sheaths speed up the action of an impulse. It can now jump from node to node instead of the standard domino effect of action potential and depolarization. This form of conduction is called saltatory conduction.